Welcome to this explainer. Today, we're diving deep into the literal energy economy of life on Earth. We're going to transform some pretty complex biochemistry into a fascinating story of, well, making bank and spending cash. Because if you think about it, every single movement you make, every thought you have, it all requires currency. And in the biological world, that currency, basically the single dollar bill of the cell, is a molecule called ATP.
So, how does a beam of sunlight, traveling millions of miles through space, eventually power every single beat of your heart? I mean, it sounds a bit like magic, right? But I promise you, by the end of this explainer, you'll understand exactly how that solar energy is captured, packaged, and spent by living cells through an incredibly precise system.
Here's our roadmap for today. We'll hit the cycle of energy, then capturing the sun's energy, adapting to the environment, glycolysis, and finally, mitochondria power.
All right, let's jump straight into Part 1, the cycle of energy, making and spending cash.
You see, biology follows this incredibly efficient biochemical economy. You know the rule, you can't spend what you haven't earned. On one side of this system, we have the chloroplast. This is where plants literally make bank by capturing light to build glucose. And on the flip side, we have the mitochondrion. That's where organisms spend cash by breaking that glucose down to extract usable energy. Really, they're just two halves of the same magnificent engine, literally breathing in what the other breathes out.
Moving right along to Part 2, capturing the sun's energy, photosynthesis.
Okay, let's look at how the money is actually made, focusing on the remarkable stuff happening right inside plant leaves. Photosynthesis, it isn't just one simple step. It actually splits into two distinct phases. First up, you've got the light-dependent reactions in the grana. These physically capture photons of light. Then, the dark reactions in the fluid stroma kick in, using that harvested chemical energy to actually build the sugar. Now, this energetic zigzag is what we call the Z-scheme. Picture this. Light hits photosystem 2, splitting water and boosting electrons to a way higher energy level. They then pass down an electron transport chain, get boosted yet again at photosystem 1, and directly fuel the production of NADPH. And that NADPH? It's a highly charged molecule that acts as totally crucial building material. So to finalize this deposit into our energy bank, we enter the Calvin cycle. And wow, think about the sheer biological cost here. This cycle has to turn a full 6 times, utilizing a massive 18 ATP and 12 NADPH just to produce one single molecule of glucose. I mean, that is a tremendous initial investment of biological capital just to lock solar energy into a stable, transportable sugar molecule.
Which brings us to Part 3, adapting to the environment with C3, C4, and CAM pathways.
Now, obviously, the natural world isn't perfect. It gets hot, it gets dry, it's totally unpredictable. Because of that, plants have had to dramatically adapt their manufacturing lines just to survive. Most plants are C3 plants, and they work wonderfully in temperate conditions. But C4 plants, think maize, they actually evolved a special structure called Cran's anatomy specifically for dry, tropical regions. And even more extreme, CAM plants, like desert cacti. These guys only open their pores at night to collect carbon dioxide, that way they don't lose precious water to the blazing sun.
So why exactly did C4 and CAM plants evolve these incredibly complex workarounds in the first place? Well, they did it specifically to avoid a huge biological mistake called photorespiration. This happens when a key enzyme, rubisco, accidentally grabs oxygen instead of carbon dioxide. And when it binds oxygen, it just wastes energy and carbon. It's a highly wasteful process that completely drains the plant's resources. So the C4 and CAM pathways are really nature's ingenious way of ensuring rubisco only ever sees carbon dioxide, keeping the whole factory running super efficiently.
Okay, let's flip the script. Part 4, glycolysis, the first breakdown.
So an animal eats a plant, and boom, it is time to cash out that stored glucose. This begins right in the fluid of the cell, the cytosol. The breakdown of glucose starts with glycolysis, which is a fundamental 10-step process. But we can simplify it into 2 distinct financial phases. First off, you literally have to spend money to make money. In the energy investment phase, that's steps 1 through 5, the cell actually spends 2 ATP to prime and split the glucose. But then, we hit the payoff phase in steps 6 through 10. That's where the cell finally starts extracting energy, generating ATP and NADH.
To kickstart this whole machinery, there's this enzyme called hexokinase, and it irreversibly traps glucose inside the cell by attaching a phosphate to it. It's pretty brilliant. By the end of this incredibly intricate 10-step cascade, that original 6-carbon sugar is split perfectly in half, which leaves us with 2 3-carbon molecules called pyruvate.
Now, what happens next depends entirely on one thing: oxygen. If you have oxygen, aerobic glycolysis feeds right into the next phase, ultimately yielding an equivalent of 7 ATP from this first part. But let's say you're sprinting hard and oxygen runs out. Well, anaerobic pathways take over. The system basically stalls out, and you're left with a net profit of merely 2 ATP. So yeah, in cellular respiration, oxygen is absolutely the key to real sustainable wealth.
Moving to our final stage, Part 5, mitochondria, powering the cell.
To unlock the full, absolute potential of that glucose, the pyruvate gets transported into the powerhouse itself. Deep inside the interior and the mitochondrial matrix, we enter the Krebs cycle. Honestly, think of this cycle as a highly complex molecular shredder. It meticulously oxidizes the carbon skeleton that's left over from the glucose. As the molecules cycle through, carbon is literally ripped off and released as the CO2 that you exhale, all while the cycle extracts high-energy electrons at multiple, very specific steps.
So for every single molecule of glucose that enters the system, the Krebs cycle yields 6 NADH, 2 FADH2, and 2 GTP. But here's the secret. The true prize here isn't the tiny bit of immediate ATP. No, the real treasure lies in those electron carriers, the NADH and FADH2. Think of them like fully loaded biochemical gift cards, just packed with potential energy waiting to be redeemed.
And finally, we reach the grand finale. Those gift cards, they get cashed in at the electron transport chain, which is physically embedded right on the inner membrane of the mitochondria. The NADH and FADH2 drop off their high-energy electrons, and as these electrons bounce down the chain, their energy is used to power these tiny molecular pumps. These pumps push protons across the membrane, creating a massive, highly pressurized proton gradient. It's exactly like water held back behind a giant biological dam.
When those protons are finally allowed to flow back across the membrane through this tiny, amazing molecular turbine called ATP synthase, the kinetic energy is used to forge brand new ATP. The result of all this? The system yields an astonishing 36 to 38 ATP per single glucose molecule. This is the ultimate energetic payload of aerobic respiration. I mean, we went from a measly 2 ATP profit without oxygen to an absolute jackpot with it.
Which brings us to a pretty mind-bending reality. Right now, as you're listening to this, every breath you take, every bite of food you eat, it fuels this incredibly intricate biochemical engine happening inside trillions of your cells simultaneously. So if a single glucose molecule holds all this power, how efficiently is your body managing its own massive energy economy right now? And it really makes you wonder, what other hidden microscopic cycles are flawlessly powering your existence without you even realizing it? Keep asking those questions and keep exploring the amazing mechanics of life.